thanks for the invitation. This is my first commitment in this academic year, and it's actually an amazing way to start it. Usually it's a graduate lecture, so this is way more fun. It's actually a very exciting time for me to be invited to give this talk, because we have a story that we just posted on my archive, and I would love to hear what you think about it. I'm gonna talk about a couple of different stories, fairly different from each other, so it's always hard for me to come up with a title that would encompass them both. So I decided to just call this talk of killers and ligands, unexpected immunology learned from cancer, because my lab and my interest has always been historically to look into immune responses to tumor, but I like to think of myself as a basic scientist, and I believe that by studying the immune response to tumors, we can learn a lot about how the immune system works. And of course, if we develop some therapies as a product of this study, it's fantastic. This is my group. This picture has been taken when we had our virtual lab party in 2021. We don't have an updated picture, unfortunately, quite shamefully. So a few people that just joined the lab. It's a fantastic group, and I would encourage you to follow the lab on Twitter if you are not. The students post quite a lot of fun things, much more than I do on my fairly boring Twitter. First of all, some disclaimers. I've been paid to consult for a number of companies, and I also have sponsored research agreements with a couple of biotechs, but none of these is going to matter for this talk. I'm not going to present any of those studies. So, as Anna suggested, as Anna mentioned, my lab is interested almost exclusively in studying the immune response to cancer, and particularly the response of natural killer cells to cancer. And we've been fairly lucky to publish a few studies on this. And then we had a little bit of a detour as a result of a fantastic student joining the lab, and that's what I'm going to focus in the second part of my talk about the oncolytic virus project. But first, let me start with the PD-1 on NK cell aspects, which is probably one of the reasons why Anna decided to invite me. This is work that we published back in 2018 and earlier this year. So I'm not going to spend too much time on it, because it's all fairly published stuff, but just a few highlights. And this work is something that I have started when I was much younger and cooler. I'm still in Berkeley. I think, in fact, Anna and I overlapped in Berkeley at the time that I was starting this study, and was mostly developed by a very talented undergraduate student at the lab at the time, Joy Su, who is now a graduate student in New York at Sloan, and by Jonathan Hodgins, who joined my lab as the first graduate student when I moved to Ottawa. And without entering too much into the details of the story, which is fairly old now, it's been 4 years, what we found was that natural killer cells have a quite suppressed phenotype in the tumor microenvironment. And at the time, it was not known whether PD-1, where of course there was a lot of interest in studying the biology of this immune receptor, was involved in any way, shape, or form into suppressing natural killer cells and natural killer cell responses. And what we found out was, in fact, that natural killer cells in a number of solid tumors, a number of mouse models, tumor mouse models, when we look in the NK cells infiltrating the tumor bed, we found a lot of PD-1 on the surface of these NK cells. Quite interestingly, the expression of PD-1, or at least the presence of PD-1 on these NK cells, did not correlate with an exhausted phenotype. In fact, these NK cells express markers of activation, which we have shown as CD69 and SCA1, but a few others as well, which to me seem to indicate that the NK cells get in tumor microenvironment, and the ones that are activated also start producing PD-1. And the expression of PD-1 is a mechanism to tune down the response of NK cells, and you always want a negative feedback to prevent NK cells from becoming autoreactive. And this is, of course, exploited by tumors. And when a tumor cell does not express PD-L1, the NK cell, even if express PD-1, cannot be suppressed, and so there is tumor killing. But on the other hand, if a tumor cell express PD-L1, the ligand for PD-1, this engagement results in some inhibitory signals that we have not characterized on the NK cells, and the suppression of the NK cell response results in escape and tumor growth, and eventually the mice would succumb out of these tumors. And if you have noticed, I have been a little bit reluctant mentioning expression of PD-1 on these cells. At least I've tried, because one thing that we left open on this paper and that we couldn't figure out for quite a while actually was how is PD-1 present on the surface of these NK cells. And since I'm a fairly simple man, the model that I was hypothesizing was going to happen was something similar to T cells. In T cells, the expression of PD-1 is mostly driven by signaling downstream of the TCR or by some environmental clues like cytokines, most pro-inflammatory cytokines, IL-2, IL-12, etc. And the exposition of the T cells to this environmental clue upregulates PD-1, and the T cells now are primed to become exhausted or at least to be suppressed by PD-1. And so I thought, for NK cells, these are fairly similar cells after all, in many ways, and it's going to be something very similar. So we start studying which activating receptors could drive the expression of PD-1, or if some inflammatory mediators, like cytokines, for example, or even suppressive mediators like glucocorticoids, etc., could drive the expression of PD-1 in NK cells. And this took me a very long time, and resulted in pretty much nothing. There was nothing in my hands that could really upregulate PD-1 in vitro, or even by conducting some ingenious in vivo studies by knocking out or knocking in molecules. We couldn't in tumor cells. We couldn't find a pair of receptor ligands or cytokine receptor signaling components that could upregulate PD-1 on the NK cells. And then we finally remembered something that was in the back of my mind for quite a while, and which is in these cancer models that we have studied are basically T-cell cancer models. These are models of leukemia. These T cells, these tumor cells, being T cells, are primed to express very high amounts of PD-1 because that's what T cells like to do. And so these T cells express PD-1, and then I remember a different study that was published back in the early 2000s and in 2000, early 2000 and late in 2010 on this process called trogocytosis, in which immune cells, including NK cells, have the capacity of capturing the membrane of cells they interact with, and in doing so, they would carry away with them parts of those membranes and the proteins that were expressed on those membranes. And so the question that we start asking is, can NK cells actually steal PD-1 from the tumor cells? In this context, what you could imagine is going to happen is that the NK cells interact with the tumor cells, and since the tumor cells express PD-1, when NK cells interact with these tumor cells and perform trogocytosis, PD-1 can be transferred from the tumor cells onto the NK cells. And this is work that has been led by two postdocs in the lab, Shahbazim and Marie Marotel, who actually trained here in France, and that we recently published in Science Advances. The key experiments that Marie and Shad have conducted, I'm just going to show you a couple, and this is one that I really like, is that Marie and Shad took this tumor cell line that we like to use, T-cell lymphoma, RMA, and they express quite a lot of PD-1 and knocked it out. So we now have a cell line that expresses or not PD-1. And we injected these cells into mice that either are sufficient for PD-1 expression or they're deficient for PD-1 expression. These are the PD-CD1 mice that Arlene Sharpie generated. And when we injected these cells, our anticipation would be that if PD-1 is expressed by NK cells, then these cohorts of mice, the one that's sufficient for PD-1, would express PD-1 regardless of the expression of the PD-1 molecule on the tumor cells. But on the other hand, if the tumor cells donate PD-1 to the NK cells, then NK cells would become positive for PD-1, even in mice that do not express, that cannot genetically express PD-1 themselves. And to our surprise, this was the really only mechanisms that led to the expressions, to the presence of PD-1 on the surface of the NK cells. If you look at this upper part of the graph, when we injected the wild-type tumor cells into the wild-type mice, we see this very nice expression of PD-1. And here, what I'm showing is PD-1 on the x-axis and tau 1.1 on the y-axis. What I forgot to mention is that we transduce these tumor cells with tau 1.1, which cannot be genetically expressed by the B6 mice, which express tau 1.2, and therefore can be used as a proxy for trogocytosis, for donating part of the membrane. The NK cells can only stain for tau 1.1 if they acquired it from the tumor cells. That's the only way that they can do that. And you can see that quite intriguingly, the cells, that was a very high PD-1 staining, but all the cells that stained for PD-1 also stained for tau 1.1. And this, interestingly, was true not only for NK cells, but also mostly for T cells. You can see here that the vast majority of cells, or at least 50% of the CD8 T cells in the tumors that stain for PD-1 also stained for tau 1.1. So this is trogocytosis is happening on both the T cells and NK cells. Now, is trogocytosis associated to PD-1 expression, or is this actually leading to the PD-1 being present on the surface of the NK cells? Well, the answers came from this group, where we injected the RMA PD-1 knockout mice, tumor cells into the wild-type mice. And if there was expression, endogenous expression of PD-1, we expect some staining, but we couldn't actually see any staining for PD-1 on the NK cells, whereas we could see that the T cells do express PD-1 endogenously as expected. On the other hand, when we injected the tumor cells that now have PD-1 into mice that cannot express PD-1, into the PD-CD1 knockout mice, we saw very similar level of PD-1 staining as compared to the mice that we injected, the wild-type mice that we injected with the same cell line, showing that the staining for PD-1 on the NK cells almost entirely, if not entirely, came from acquisition from the tumor cells via trogocytosis. And of course, when we did a complete PD-1, lack of PD-1 expression on both the tumor cells and the mice, we didn't see any staining, indicating that our staining was very clean. So this was quite surprising, and we did a bit of molecular studies to figure out where this was actually trogocytosis. We did microscopy, subtransfer, etc., transfer, and it was trogocytosis. Not gonna show you this as is all published, but something I want to point your attention towards is what is actually the functional relevance of PD-1 transfer on NK cells, because you know that immune receptors to signal the presence of the receptor by itself is not sufficient to drive signaling. There has to be clustering, there has to be a certain lipid composition on the membrane, the adapter molecules has to be there. And so the question is whether PD-1 transfer was just something that randomly happened and did not have any consequences really was bugging us. To answer this question, what we did were some in vivo experiments where we took PD-1 knockout mice and injected them mostly with RMAs expressing PD-L1. This cell line is very similar to RMA. It's just a couple of differences. One is that they do not have class 1, and so they cannot be killed by T cells. They're very refractory to T cell killing, but lack of class 1 also makes them very good target for NK cells, and that's pretty advantageous for us to study NK cell responses. And we had to transduce these cells for PD-L1 to make sure that they were able to provide inhibitory signals to the NK cells via PD-1, and this is what we published in the first JCI paper. And then we either treated the mice with PD-1 or with ice-pipe control. Before doing that, we actually did a different experiment where we took the RMAs PD-L1 and knocked out PD-1 and confirmed that there was no transfer of PD-1 in this model as well. And interestingly, these cells actually grow faster. So even in the absence of therapy, PD-1 here is actually able to suppress the NK cells, as this is the only source of PD-1 is coming from the RMA into the PD-1 knockout mice, and we showed that this is NK cell dependent. But then the interesting part was that we treated the mice with the therapeutic antibody. The mice that received the control IG grow these very nice tumors, as you would expect. But now when we treated with PD-1 blockade, the antibody was able to prevent the inhibition of the NK cells. And as you can see in this group here, the growth is, of the tumors is greatly reduced, indicating that the PD-1 that has been transferred to the NK cells from the tumor cells is actually able to signal and function and to suppress the NK cell response in quite a dramatic fashion, because both these experiments would indicate that PD-1 is greatly contributing to suppressing NK cells in the tumor microenvironment. So in conclusion, I did not have time to show you all the data, because I want to move to the next part of my talk, where I am hopefully going to be a little bit more thorough. What we show is that a bunch of immune cells, and I just show you T cells and NK cells, but we really looked into a number of other immune cells, and all of them can perform trogocytosis at very high rates in cancer cells. And this is known. We already knew that this was happening. There's some beautiful studies published a couple of decades ago now have shown this. What we added to the conversation here was that actually trogocytosis, it's a very important mechanism by which tumor cells try to make the immune system more tolerant to its presence. And they do so by donating molecules that could potentially suppress the immune response. And in this case, we studied PD-1, but it's not impossible to exclude that other molecules can be transferred from some other tumors into immune cells. And these other molecules could play very similar roles to the one that we have identified for PD-1. We did some molecular studies and showed that actually the SLAM receptors, this massive family of receptors, are essential for trogocytosis. So they knock out these genes. You cannot see any trogocytosis, which has some very important implications in the clinic as well, because some SLAM receptors are actually be targeted by monoclonal antibody therapies. And trogocytosis can inhibit PD-1. Trogocytose PD-1 can inhibit anti-cancer immunity, and we actually have evidence that this is happening in human patients. We were lucky enough to collaborate with a fantastic hematologist here at the Ottawa Hospital, Ardolino, and we studied a cohort of myeloma patients, multiple myeloma patients, and found indication that PD-1 was also transferred from the myeloma cells into the NK cells and potentially T cells. We had a number of questions that we need to answer, of course. For example, is the signaling of PD-1 in trogocytosis the same that we would expect? And what's the strength of the signaling? The in vivo experiments suggest it's pretty strong, but we need to show that. How does actually PD-1 suppress the NK cells? This is a question that has eluded my understanding for the longer time, because even in the first JCI paper, when we show for the first time that PD-1 can suppress NK cells, the molecular details of how this happened, we are not so good at figuring it out. And I think part of the reason was that this is due to trogocytosis, and so it could be slightly different than a molecule that it's expressed. And so we expect that maybe there's something else going on. Of course, can we therapeutically block trogocytosis? This is a very important question, because as I mentioned before, if this is a common mechanism by which tumors can adapt the immune system, can suppress the immune system, then if we have a drug or if we have a therapeutic that we can use to prevent trogocytosis, maybe we block broadly a number of single molecules that targeting individually would take a great effort. So we're moving on with the studies, and this is my daughter helping me out writing a grant to actually try to move on with this. And if you're interested in this, we most likely are going to be recruiting people for keep on this what I think pretty interesting study. So if you want to talk to me about it, I'd be very happy. So now let me switch gear quite dramatically and talk about this preprint that we just deposited. And I should start mentioning that there's no NK cells here. So this is way out of my deep, and I should acknowledge that this work has been both experimentally and intellectually greatly driven by Jonathan Olchins, who is a fantastic graduate student. He is about to graduate and is going to move to Genentech by at the end of the year. Fortunately for me, very very fortunately for him, but he was really a true force of nature in conducting this study. Because again, my lab has no expertise in oncolytic viruses and metabolic shifts or in type 1 interferon. And so pretty much every question that he had, I had to just answer, well, you gotta figure it out yourself. And he did. He did a very, very excellent job in this. He was, of course, not working alone. He was helped by many people. I'm going to try to mention them during the rest of the talk. Two people worth mentioning: Maria Park, a very good, very great undergraduate student to work in the lab for a couple of years, and Ash Agerman, who is now, she's in med school now, unfortunately, and Ash Agerman, who is a fantastic undergraduate student, who is now a graduate student at UOT and the University of Toronto, and they're lucky to have her. And just a little bit of introduction of why we decided to take on this was that PD-1, as you surely know, and I skimmed through this very quickly in the first part of my talk, but it's a major cancer. It is a major target of cancer immunotherapy, and these are data showing the effectiveness of atezolizumab, who is the monoclonal targeting PD-1 in combination in side-by-side comparison with chemo. And you can see that tumors that express high amounts of PD-1, atezolizumab does much better than chemotherapy. And it's great, as most immunotherapy are, particularly if you stratify it, but the response is not complete. There's a lot of room for improvement. The overall survival is about 50%, which again, fantastic, but these other people need definitely a alternative treatment. And what we think, and believe that the field is moving in this direction, is that the response is, so the answer to this is going to be combination of therapies. We're going to be able to combine different immunotherapies potentially to prevent adaptation, tumor adaptation, or even lack of response to begin with to the first immunotherapy. And one thing that we really like, I'm actually think I'm developing a cold. So one thing that we really like here in Ottawa is to study oncolytic viruses, and this is because of the effort led by my colleague John Bell, who was one of the pioneers in the field. And if you're not familiar with this, the concept behind oncolytic viruses, that these are viruses that naturally or we have engineered to not be able to productively infect healthy cells. Healthy cells tend to have very good interferon response or other antiviral measurements, but mostly interferon response. And so when a crappy virus infects them, they can get rid of it quite nicely. On the other hand, tumor cells, while they evolve and become tumor cells, when they transform, they very often lose the ability of responding to viral infection. Mutations in the interferon pathways are very often associated with the tumorigenesis process. And it's not entirely clear whether this is just a random thing that happens due to the high rate of mutagenesis, or if there is actually a specific indication. And actually, the studies that were published recently showing that this could actually not be just something random, but for tumor cell is very important to lose that kind of response. And so when a tumor cell that doesn't have interferon is exposed to oncolytic viruses, what happens is that the healthy cell can get rid of it, tumor cells cannot get rid of it, and actually there is viral replication and tumor cell lysis. And now you have a therapeutic, a biotherapeutic, which cannot harm the healthy cells around the tumors, but can in fact kill the tumor cells. And this was the idea, initial idea behind oncolytic viruses, was that you would have an agent that would directly kill in a more specific fashion than chemotherapy, and potentially in a less invasive and damaging for the patient fashion than the chemotherapy. The tumor cells. And then, more studies, of course, have shown that as for any kind of therapy, really, the key here is the immune system. And in fact, a big part of the therapeutic response of oncolytic viruses is driven by the immune response that is elicited by the virus in many different ways that are not relevant to this talk. And so, of course, this was amazing news, and the development of oncolytic viruses has proceeded very, very quickly. And in fact, there's an oncolytic virus that's been approved, TVAC, in melanoma patients. This is HSV1 expressed in GM-CSF, which matters us only very little. But the one thing that it's actually very important to notice is that virtually all clinical trials that have been conducted in the clinic have some sort of arms where the oncolytic virus is combined with other immunotherapies, and most often these other immunotherapies are blockade of the PD-1, PD-L1 axis, because we really like to do that. It's a very powerful axis. It suppresses T cells very strongly, so we want that to do. And in the PD-L1 blockade plus oncolytic virus, there are 8 now that are recruiting, and this slide is about a year old now. I didn't update it, but probably there are more right now. And actually, the group of Zamarin published a very nice paper in JCI back in 2018, showing that combination of a different oncolytic virus, the Newcastle disease virus, with anti-PD-1 or anti-PD-L1, very greatly improved the survival of mice injected with some tumors, whereas the single agents didn't work so nicely. So there's a lot of room for combination, and there's of course a lot of interest in this combination, not just in preclinical research but in the clinic as well. But what has not been entirely studied, or at all studied, I should say, this is where Jonathan kicks in here, is whether the two pathways, the PD-1 and PD-L1 axis and the oncolytic virus axis here, are interdependent with each other, and whether tampering with one axis could potentially affect one other axis. And this is an interesting question that was mostly raised by some very nice studies that have been showing that PD-L1 is much more than a ligand. We actually called it PD-L1 as the PD-1 ligand number 1, as it was identified, but this is just a nomenclature, and is really due to our way of studying immunology, which is very T-cell centric. And so if the receptor is expressed on T cells and something else binds that receptor, most likely that's just a ligand. It turns out that's not really the case. PD-L1 has a number of other functions that are really PD-1 independent, at least in terms of cell intrinsic effect on the cell that expresses PD-L1. And this is true both for tumor cells and many immune cells. And these papers have been published fairly recently. I believe that they started around 2017 or so. What I'm showing here is most recent papers published from 2020 to 2022, give or take. And there's a fair amount of literature now showing that PD-L1 has some intrinsic effect. This is just a slide from 2018 suggesting that PD-L1 can act as an oncogene, and these are some of the pathways that could potentially lead to its role as an oncogene. And it's not the reason why I'm pointing this out is just to try to convince you that there is solid literature now showing that PD-L1 does not just bind to PD-1, but has a cell intrinsic effect on the cell that does express PD-L1. And so the question was, could PD-L1 signaling regulate the sensitivity to oncolytic viruses in a model of interest? The model of interest that we decided to pick was the prostate cancer model. We really like this cancer cell line called TRMP2. The reason why we started to study this prostate cancer model is because oncolytic viruses are in the clinic for prostate cancer, and there is a lot of interest. Of course, there's a lot of room for improvement of immunotherapies in prostate cancer, and there are reasons to believe that oncolytic viruses could actually work very nicely in this indication. But then there's not much mechanistic studies. That's why we wanted to figure this out when we started the work. And we decided to use this virus called VSV Delta 51, which is a virus that was already available in the lab, as was developed by the group of John. This is a single-strand RNA virus. It can infect mammalian cells by binding to the LDL receptor, but the mutation in this protein called M51 makes these cells this virus extremely sensitive to type 1 interferon. A very little amount of type 1 interferon will make this virus not be able to infect these cells any cell really very well. And so this is a great model, we like to think, because these cells, we need a tool to study the immunotherapeutic response in prostate cancer, and we have a very good virus to study this. This is oncolytic, so when it gets into the cancer cells, it kills them, and it can elicit an immune response. And this is at least what we decided that we wanted to study, the immune response in response to the oncolytic virus. But then Jordan one day showed up in my office with this particular picture, and I'm showing you this as indication of how dramatic the phenotype here was. What Jordan did was to take these TRPC2 cells, and what is common in the field is just to infect them in a dish to try to figure out what the sensitivity of the cells is. And we knew that these cells were sensitive to oncolytic viruses. We are not the first one infecting them. And what you can see here is the increasing amount of viruses that we added to the cells. And you see that even a very small amount of virus can basically blast the cells off. What you see in blue is cells that remain alive in suspension, adherent to a plate. This is co-massive staining. But you see even at the 0.001, so this is one particle of virus every 1,000 cells, you can see that the cells are basically all gone, and they are actually all gone from 0.1, I would argue, probably. So anyways, very high sensitivity. And this cell, just because we could, we decided to knock out PD-1 for different reasons, for different studies. And Jordan had these cells in his hands, and was interested in trying to figure out whether PD-1 could play a role here, and decided to infect the TRPC2 PD-1 knockout cells as well with the oncolytic viruses. And what he saw, very dramatic phenotype, was that the cells that now lack PD-1 expression became incredibly resistant to the virus. You can see here at 0.001, there is no cells that remain alive basically in the TRPC2, but in the TRPC2 PD-1, they're all alive. And we start seeing evident signs of that probably at the 1 MOI and not 10 MOI. The cells start lifting up, but this is a very high MOI that, it's kind of super physiological, really. So there was a great extent of resistance that was conferred when we knocked out PD-1. And of course, I asked Jonathan to repeat this experiment 75 times, really, because I was very skeptical about this, and I thought that he was just messing something up. And he tried with different batches of cells, with different viruses. He actually subcloned the cells in different clones, such that we could see whether this is a specific thing or it was widespread, and everything worked out. This actually is a real phenotype. Every time that we repeat this experiment, we saw these dramatic differences. And virus people really like this assay called Blake assay, which we have done quite diligently in the majority of the experiments that I'm going to show you. And this assay is used to quantify the actual amount of virus after infection. As you can see here, in the blue, you will have always the TRPC2, and in the red, you will always have the cells lacking PD-1 expression. When we infected these cells with these two cells with the virus, the parental cells, the one that express PD-1, are greatly more sensitive to the infection. You see always higher amounts of virus in this cell line. And for the majority of the experiments, actually, I'm going to show data that we did by flow, because we are an immunology lab after all, so we do like flow very much. So we have a virus that express YFP as a reporter, and when we infect the cells become YFP positive, and we can very quickly stain for them, well, not even stain, just flow them, and see the amount of virus, the percentage of cells that got infected with the virus. And this data corroborate this data very nicely when you can see that there is no infection, there's no YFP signal, but with increasing amount of infection, there is increasing amount of the signal on the tumor cells. And this was much less dramatic in the cells that have lost when we knocked out PD-1 expression. You can see signal really only at the 10 MOI, consistent to the previous data. Of course, plaque acids are more sensitive, but all the data that we have with YFP reporter were corroborated with plaque acids. And of course, the phenotype was rescued by PD-1 recomplementation. When we added PD-1 back to the TRPC2, they really look like to the TRPC2 PD-1 knockout, they really look like the parental cells. So this was not just the experimental artifact as a result of PD-1 being knocked out by CRISPR-Cas9, but it was really due to the expression of PD-1. And so is this really signaling? And we don't know how PD-1 signal. That's one direction that we have to take, but it's very hard for us to figure out what's going on inside the cell. But an indication that we can take is that if this is really cell signaling, we would argue probably a ligand has to be provided in some way, shape, or form. Or if you actually trigger the receptor, in this case PD-1, you should be able to get higher phenotypic effects. And so what Jonathan did was stain for the TRMC2 cells for the two known mouse ligands of PD-L1, which are PD-1, and it's of course not expressed by this prostate cancer cell line, as you wouldn't expect, and CD80, which is actually very interestingly expressed only by half of the tumor cells, roughly. And this is not stable. You know, if you put them back in culture and give enough time, it will go back to this phenotype. But you know, in culture, you have the heterogeneity of CD80 expression. So Jonathan sorted these guys and these guys. For whatever reason, we couldn't knock out CD80 on these cells, and I believe there might be a couple of reasons why that's the case. But you know, it's beyond the scope of this talk. And of course, when we take the CD80 positive cells, we are hypothesizing that these cells are able to, CD80 is able to bind to PD-L1 either in cis or in trans, and this binding would result to the phenotype. And in fact, we saw very similar to what I've shown you with the cells that with the bulk population. There was a very nice difference in infectivity between the TRMC2 and the TRMC2 lacking PD-L1 expression. The blue bars and the red bars are very similar to the ones in the previous graph that I showed you. But when we took the CD80 knockout cells, so these cells don't have CD80, so there's arguably no cellular ligand for PD-L1, then the effect and the differences were completely lost. You can see that the resistance of the PD-L1 knockout cell was gone, and they infected as equally as well as the parental cell line, indicating that CD80 is required. The binding of CD80 to PD-L1 is required to provide the inhibitory signal. Sorry, the signal that is triggering resistance. Sensitivity, sorry, to these viruses. Is this specific for CD80, or if PD-1 is around, does it matter? We asked the question, and we used the PD-1 fusion protein and triggered PD-L1 with this fusion protein, and actually we saw an increase in infection in the cells that express PD-L1, suggesting that in this system there is no PD-1 available, but if there was PD-1 available, then you have the ability of triggering the very same signal. And I should mention CD80 and PD-1 are known to bind to PD-L1 in non-competing fashion. So the same molecule can bind to both CD80. The same PD-L1 molecule can bind to both CD80 and PD-1. So it's not surprising that you have this effect, and then you have PD-1 right here, and you see a further boost because there is even more signaling coming down that route. And then, of course, we ask the obvious question: if we use monoclonal antibodies, which are widely used in the clinic and in preclinical model, can we mimic the same signal? And in vitro, we definitely can. We have 3 clones that we got from Ira Mellman at Genentech, and these clones bind to either CD80, mimic the binding of CD80 or PD-1 to PD-L1. And what we saw with the clones that mimic the binding of PD-1, we saw a very nice increase in infection in the cells pre-treated with these clones, but that was not the case when we pre-treated the cells with clones that only mimic the binding of CD80, because this CD80 was already binding to PD-L1, and so if you have an antibody, nothing new is going to happen. And of course, in the knockout, we didn't see any, in the PD-L1 knockout, we didn't see any effect, showing that this actually was driven by PD-L1. This also alleviates our concern that potentially some CD80 signaling could be driving the phenotype and not PD-L1 signaling, because we are actually, in this case, and as in the previous experiment, directly triggering PD-L1 with an antibody or a fusion protein. So this worked out great for us. And then we asked the question, and then at this point we have this phenotype where PD-L1 is linked to our susceptibility to oncolytic viruses, but we don't know how this is happening, right? How does PD-L1 favor infection? And the answer to this question partially came from this very nice paper published by David Ascor back in 2017, where Jonathan was starting the study, showing that in a different cellular model, PD-L1 is able to suppress interferon, signaling downstream the interferon receptor. And of course, interference is very important in our context. And so we did the very same line of studies and showed that, in fact, PD-L1 presence resulted in a higher expression, in a lower expression of interferon, both at the transcript and at the protein level. And so when we knocked out PD-1, we saw higher responses of interferon in response to the expression of interferon in response to the virus. And we treated the cells with the PD-1 antibody, actually decreased the expression of type 1 interferon, showing that this actually was the case. And we have a bunch of experiments there in the paper showing that this is actually the pathway that is very important here. The cool thing that we actually found out was that it was not something that happened exclusively after oncolytic virus infection, but was in fact something that was imprinted in the cancer cell line. There was in fact the basal responsiveness of type 1 interferon that was lower in the cells that express PD-1. And this is very interesting for evolutionary reasons, because PD-1 is one of the major targets of type 1 interferon. When there is type 1 interferon, PD-1 is upregulated by virtually every cell, really. And you can think that if PD-1 signaling is suppressing type 1 interferon, that could be a mechanism that has evolved for PD-1 to suppress a negative feedback loop, for PD-1 to suppress the reason why it has been brought up type 1 interferon. And you want to prevent type 1 interferon from causing autoimmunity. So in this case, PD-1 is actually very important to prevent autoimmunity, not only by regulating T cells directly via engagement of PD-1, but also by, in a cell-intrinsic fashion, suppressing the expression of type 1 interferon. And this is some very nice evolutionary mechanism that I really enjoy. So we know that PD-1 goes through type 1 interferon, and that results in a higher susceptibility to oncolytic viruses. But how does this happen, right? We don't know the link between these two events. And so this is when collaborations with this fantastic graduate student in the lab, Bart van der Eyden, kicked in. He's a bioinformatician, and so we did some RNA-seq, and he analyzed the data with this very cool tool called Progeny, where you can actually take not just single genes, but the whole pathways. And these old pathways, you can see whether or not they are more or less active in the cells of interest. And we saw that in our cells before infection, so without any virus, 6 pathways were more active in the PD-1 expressing cells. And one pathway, the TGF, was less active in the PD-1 expressing cells after infection, but not before infection. We did a lot of pharmacological experiments, and none of these pathways turned out to be any relevant for our phenotype. We didn't investigate P53, but knocking on woods, that's not very important. What turned out to be very important was this hypoxia pathway. And when we looked at a closer look at the hypoxia pathway, what we found out was that this hypoxia pathway was not really just hypoxia, but was very tightly linked to metabolic effects. And in fact, when we looked at when we analyzed this data in a different way with this glycolysis gene SAT score, which indicates how much of these glycolytic genes and consequences of this gene expression are brought up in the Q cell lines, we saw quite a steeper increase, in the TRMC2 cells than the TRMC2 PD-1 knockout. At this point, I was way out of my reach. I don't know anything about metabolism. I used to love it when I was an undergraduate student, but then I lost track of it, and so that was. We were in big trouble, but fortunately, when you're in big trouble here and you pick up the phone, you have a number of fantastic colleagues that you can kind of exploit, really, to establish very nice collaborations that benefited my lab immensely. And one of these collaborations was with the lab of Michele at the University of Ottawa, and this was work that was done also by its excellent student, Claire von McMaster, and they helped us performing some seahorse assay, which help us determining the bioenergetic production of the where the ATP is coming from, and whether glycolysis or OXPHOS are more involved into the processes of energy production. And we could see that actually measuring glycolysis, I don't want to enter into the technicality of this too much, it doesn't really matter. The cells that express PD-1 have a higher rate of glycolysis. This is basically what it's indicating. And in fact, the production of ATP in the cells that express PD-1 was more skewed towards the glycolysis than OXPHOS, which indicates that these cells are doing a lot of glycolysis. We wanted to know whether this was actually true in vivo as well. And that's where help from the group of Beth Rothstein at the Art Institute here in Ottawa came very handy. And they have this fantastic thing where they can do PET scanning to mice. And if you know about PET scan, what PET scan is measuring is basically glucose uptake into cells, and tumor cells like to uptake glucose much more than healthy cells, and that's why it's used for diagnostic purposes. But in our end, what we did was to inject tumor cells that either express or not PD-1 into mice that don't have an immune system, of course, and so that the cells that express PD-1 uptake glucose much more strongly. It's this very bright band here, very bright image here over the mice that don't express PD-1, and this was mimicked in vitro, by just a very traditional glucose uptake assay. And higher glycolysis and higher glucose uptake are what cancer cells like to do when they need to grow, and they do this thing called Warburg metabolism, where the OXPHOS is not so much exploited for producing ATP, but ATP is produced quickly via either aerobic or anaerobic glycolysis, and a lot of the ATP comes from those pathways. So we now know that PD-1 is linked with glycolysis in some way, shape, or form, which was not entirely known before. And there is less interferon here, but we don't know how these things link with each other. But before we took a step back and start asking, is this true only in our cell line, or is actually a more broad phenotype? And we would argue this is probably a more broad phenotype because PD-1 is such widely expressed. And so what this other fantastic graduate student in the lab that helped us doing was to take 600 cell lines, human cell lines that have been sequenced, RNA-seqed, and analyze the PD-1 expression with the glycolysis gene set score, similar to what David did in our previous experiment. And you see a very nice correlation here with between the expression of PD-1 and the glycolysis score. And this correlation doesn't mean much because one can be linked to the other. So, but what's actually very interesting for us is that we now can rationally identify cell lines where we would expect PD-1 to be able to suppress type 1 interferon, or at least to increase metabolism if these features were conserved as they were in the TROMC2 model. And we picked these two cell line HS746 and 786O because they were already available in the lab, and they were in two different quadrants of this graph. And so that, in fact, knocking out PD-1 in these two human cell lines resulted in very similar effect. The viruses were, the euclidic virus was much better infecting the parental cells over the PD-1 knockout. And quite interestingly, the phenotype was way more dramatic in the cells that express a lot of PD-1 and a lot of PD-1 over the cells that express less PD-1. This was also somehow corroborated in a data set of single-cell RNA-seq of 266 human tumors of different origins. You can see that the average glycolysis score and the PD-1 expression are plotted here. And this graph looks very similar to this graph. And in this graph, we're only plotting the tumor cells, not the immune cell component. So this could be actually true in people as well. So we now kind of think that we have PD-1 glycolysis, less interferon, and higher susceptibility on oncolytic viruses. How do these things tie together? We didn't know until this paper was published, where that in exacerbating glycolysis, there's not enough time for pyruvate to go into the Krebs cycle, and in fact, pyruvate is converted into lactate because you need back the NAD plus to redo the oxidation of glucose. And so this lactate has, for the longest time, been considered a byproduct of high glycolysis, but in fact, it has a lot of biological relevance and partakes in a lot of biological processes, one of which has been shown a few years ago by the group of Lean, is suppressing signaling of MABs and LRR, and these molecules are extremely important for the production of the type 1 interferon. And so the question that we start asking is lactate production actually responsible for their susceptibility to oncolytic viruses that we observed in our ants? And one indication that this was the case was that actually more lactate was produced in PD-1 expressing cells. You can see the difference is not that dramatic in TROMC2, but are really dramatic in these human cell lines. And so what we did was tampering with the amount of lactate that was present in the cells, and we either blocked lactate production or added lactate back, or did a bunch of other experiments really. And what we show was that by tampering with lactate production, you also alter the sensitivity to oncolytic viruses, and by result also the production of interference, which I'm not showing you here, in a way that would suggest lactate is actually the thing that is suppressing type 1 interferon when PD-1 was present. So now we have PD-1, there is more glycolysis, and this more glycolysis leads to lactate and less interferon, and so you have higher susceptibility to oncolytic viruses. Cool biology, but do we have to care about this? I would argue yes. First of all, because it's cool. Second of all, because actually it could have some very nice therapeutic implications. We took NSG mice, which don't have immune system, mostly because we wanted a system where there was no PD-1 around that could mess up the interpretation of the experiment. And we inject them with either cells that express PD-1 or can't, and then infect them with the virus. And you can see that the replication of the virus was much more increased in the parental cell over the PD-1 knockout cell line. We also did the experiment where we treated in vitro, the human cell line with atezolizumab, which is the monoclonal antibody used in therapy for PD-1 blockade, and we saw that there was actually a steep increase in the infection of these two cell lines in response to the PD-1 triggering. And finally, what we did in collaboration with actually my boss, Rebecca Auer, who is a fantastic surgeon with a great interest in cancer research, was to collect a number of tumors from patients with different tumor indications, and in vitro infect them with oncolytic viruses. And on the side, we also were able to stain for the expression of PD-1. And we could actually see that there's a very decent, I would say, decent stratification with tumors that express PD-1 are on average infected more highly than tumors that do not express PD-1 by the oncolytic viruses, showing that this could actually be not just an interesting mechanism, but also could be used as a biomarker for, if refined, of course, for deciding which patients we want to treat with oncolytic viruses. So in conclusion, I hope I convinced you that PD-1 is here for not just binding to PD-1, but for a number of different reasons. And one reason why PD-1 is brought up is because any kind of insult can lead to interferon, and interferon leads to PD-1 upregulation. If the consequence of this is that there is more glycolysis and there's warmer metabolism with mechanisms that we have not identified yet, this could lead to lactate production suppressed by point interferon as a negative feedback loop. And as I mentioned before, this evolutionary speaking is a very interesting way by which PD-1 could prevent inflammation, control inflammation in different ways that was indicated in the past. Of course, many thanks to all my lab, which I showed a picture before, all the lab members, which I believe I mentioned in all the talk. Some of the people who helped us out and remain honorary lab members over the years. It's our fantastic group at the OHRI. It's a group of about 10 scientists. We work together very nicely, otherwise actually a great place to work and to establish collaborations. University of Ottawa has been incredibly supportive, and people there just corridor away really, but great collaborators. Of course, parts of this work has been done in David's lab when I still was a postdoc. I got help from a number of people, too many to mention here, for both studies, and we couldn't have done this alone, of course. And the funding agency who helped out actually conducting the study, giving money. And something I should mention is that we're always interested in hearing new people who want to join the lab. Ottawa is a beautiful city, particularly in the winter, I would argue, when it's cold, but it's really nice, particularly it's very family friendly city. But for young people, there's a bunch of things to do, and there's a lot of nature. And if you are open to the idea, I'm happy to talk about this offline. When the visions around you